Okay. Good morning, and I welcome to both of you to our very interesting course, BTL 611 Probiotics, Prebiotics, and Postbiotics. And this is your first class, and due to COVID-19 pandemic, we are now adapted to a new world, virtual world. So this virtual classroom, I think our students, meanwhile, adapted well to this system. And virtual system has lots of advantages, and there are some limitations also. But anyway, as only two students uh, have registered in this course, I am sure we shall have enough room for interactions. So in case of undergrad student, I had 94 students online, and it was hard for me to follow them and interact with all of them. And this is why I always ask student to make another, a supplementary classroom, that is Facebook group. Uh, are you included in the Facebook group for BTL 611?
Yes, sir. I am also included.
Okay. I should thank Ruhul Amin, because you was not also familiar to open such a group. So that Facebook group is a private digital platform. So the one we are using now is synchronous. That means live. We are interacting. But in that case, you can use asynchronous communication. If you have any question, you can put there, and you can interact with your classmate, as well as you can ask question that I can answer. So we can make that page also very interactive, and I shall put the lecture and many other instructions over there. And if you find something very interesting, and nowadays internet is a global library. Lots of literatures are now available in the internet in different websites. Even in your topic, probiotics, prebiotics, and postbiotics, you can find lots of lectures, lots of publication, lots of video clips, even in the YouTube. So you can share and you can make fun and engage yourself to learn better. Ruhul Amin, do you know Sorwardi well? Sorardi, please introduce your background, because this is our first class. We should not take too much stress. And in my classroom, I always try to offer a room for the students so that they can feel comfort and enjoy learning.
Sir, shall I start?
Yes, please.
Okay, sir. My name is Hossein Sohrawardi. You can also call me Shafi because Shafi is my nickname. And also I obtained my BSc in agriculture degree from Khulna University. After that, I obtained my master's in biotechnology from Bangladesh Agricultural University. During that time, I worked in Bina, and in that time, I got some basics about um, molecular biology, especially for the practical work. And then I obtained my another master's degree from uh, George August Göttingen University, and the subject was international sustainable international agriculture. After that, I came back to my home country. Now I'm working as a plant biotechnologist in Laltir Seed Limited, and I'm acting as a acting in charge of biotech lab.
Excellent, Shafi. Really, you have diversified experiences. Khulna University, Bangladesh Agricultural University, and your
Yeah.
told your August Universität, that is the University of Göttingen. University of Göttingen, I have lots of sweet memories. I have been there for 2 years as an Alexander von Humboldt fellow. Really an amazing university in Europe. More than 55 people received Nobel Prize who are attached to that university, and the birthplace of Gauss, mathematician Gauss, and many big guys, even Einstein, Heisenberg, Robert Koch, very big guys. They worked there, and all the city roads are named after the Nobel laureates. Really, really amazing place. So I welcome you, and I hope you will enjoy similar diversified experience at our Institute of Biotechnology and Genetic Engineering, because since its beginning as Department of Biotechnology, when I joined at in 2010, I focused diversity. Here you can see Ruhul Amin. He is a completely different discipline from us. Ruhul Amin, please introduce yourself.
Uh, Assalamu alaikum. Can you hear, sir?
Yes.
Emma. Oh, Assalamu alaikum. I'm Ruhul
Walaikum
Amin.
salam.
I have been completed my college from Brahmanbaria Government College in 2013, and after that, I got an opportunity to admit into Bangabandhu Sheikh Mujibur Rahman Agricultural University, and I've taken veterinary science for my graduation, and in 2019 I have completed my graduation and in Doctor of Veterinary Medicine, and after that I it seems to me that science is going fast, and if I become an part of the molecular work or the advanced technology, it will be helpful, and that's why I have choice Institute of Biotechnology and Genetic Engineering for my postgraduate and masters, and I hope that I will be able to learn the latest technology and molecular work both theoretically and practically, and under the guidance of to very uh, renowned scientist, and I hope to and I secure dua and wish and cooperation guidelines uh, throughout my master's journey. Thank you so much.
Excellent. Ruhul Amin did his HSC from the same college. I also attended
Yes, sir.
Brahmanbaria Government College. And Ruhul Amin is a bit different discipline, that is veterinary medicine. And he's the first student from that discipline to our Institute of Biotechnology and Genetic Engineering. When I designed the ordinance of Institute of Biotechnology and Genetic Engineering, I included four disciplines. One is plant biotechnology, another microbial and industrial biotechnology, a third one fish biotechnology, and fourth one veterinary and animal biotechnology. So he is the first man and first student who started veterinary and animal biotechnology. So I welcome both of you, and diversity is the big asset in biotechnology, because biotechnology has no border. And our institute is a new institute which was established in April 2019, and it is still young, and we are still expanding and improving. So I believe both of your participation would enrich our community. We have a small community, and obviously, I am expecting you will deliver your highest level of intellectual capacity to contribute to this growing institute. from my side, I always love to encourage and support the young people, even who are beyond the field of agriculture. You are both from the agriculture discipline. I am hugely connected to a global scientific community as well as national scientific community, even those who are working outside the field of agricultural sciences, because biotechnology is borderless. When you have a DNA, then a PCR would not care whether it comes from a plant or animal or fish or anything else, or human. So hopefully uh, you will also carry such a broad mind and keep your mind open to learn new things. For example, the course BTL uh, 611, which you are going to start, uh, probiotics, prebiotics, and postbiotics. This is a unique course you cannot find in any university in Bangladesh. I also introduced some other course like
That's why
nanobiology.
I've selected this.
Yes.
I have find a different thing, and this is the time demanding course for the new century.
Thank you. Yes, it is obviously time demanding, and biotechnology, as a biotechnologist, we have to evolve in a way so that we can satisfy the demand of the society. So, probiotics, prebiotics, and postbiotics. Have you heard about these three terms? Because three keywords are included in the course title. I would like to know you whether you are familiar to.
Oh, sir, I'm familiar to the term probiotics since during my undergraduate in dairy microbiology, there are so many bacteria in milk which can help us to maintain our human gut microflora, then pH, and they also exert some beneficial impact. And I'm just introduced to the term probiotics. And last day I have also visited your um, articles in the Facebook that John Roberts, who achieved a Nobel Prize in uh, probiotics, more mm, maybe in 2005, and this type of bacteria not only may present in plant but also may present in the wildlife or our surroundings.
Excellent, excellent. So you are very familiar to the probiotics, and your concept, I think, almost clear. And probiotics, not only in the eukaryotic organism like human, you would be wondered to know that in human uh, recent human microbiome project discovered that an adult human carry 3 to 6 pounds of microorganisms. 3 to 6 pounds of microorganisms. And if you consider the number, it is more than 100 trillion. 100 trillion cells of microorganisms are living in and on our body, and we cannot sense actually, physically we cannot see them because they are microscopic, but they are incredible. Not only that, the human genome project discovered that 33%, about 33% of our genes, that is in the whole genome sequence of human, 33% area sequence is covered by the microbial genes from the microbial origin. That means over the years of co-evolution of these beneficial microorganisms with human, through horizontal gene transfer, we achieved lots of gene from the microorganisms. And probiotic microorganisms are really playing uh, very big and robust role in our growth, development, and immunity. This is why this subject is now becoming very very important, because in the past it was very difficult to estimate how many microorganisms and how many kinds of microorganisms we are carrying. But recent metagenomic approaches uh, uh, helped us to elucidate that an adult human carry up to 150 species of microorganism, and not only human. The story is not with only human. All eukaryotic organisms, including animals, including birds, including plants, all the harbor diverse microorganisms, and those who are exerting beneficial effects to the host, and those who evolved over a longer period of time, and they are called probiotic microorganisms, and mostly they are bacteria and archaea. But there are a few percentage of uh, maybe 0.001% of microorganism in the environment, they are harmful and pathogenic. So most of the microorganisms we are carrying are available in the environment. They are beneficial for the eukaryotic organisms. Okay, probiotic, I think, Shafi, what do you think? Do you like to add any point on your perception about the probiotics?
I want to add only one thing, especially for at a certain amount of bacteria and fungus, especially bacteria, because uh, especially in a limit, because we have in an optimum condition.
Mm-hmm.
Especially everything is correct in case of ruhulamin, and I want to add only one thing. This thing is because um, uh, it's a probiotic is also should be in optimum level.
Yes, obviously. Probiotics naturally there is a like homeostasis. How many probiotic microorganism uh, uh, would be colonized or living on a natural organism in the natural system. The number is optimized because they have the competition for space, competition for the food among themselves. So in case of beneficial probiotic bacteria, nowadays in many countries they are available as capsule or tablet. So probiotic capsule, one capsule contain 2 billions of bacteria, for example, lactobacillus. Lactobacillus and bifidobacteria in our gut or intestine, they are very powerful and helping us to digest the food and as well as to keep us healthy. That means boost up our immunity. Even they trigger the expression of the genes responsible for the growth and development. Even they also regulate our health, whether we will be obese or we will be not obese. Okay, probiotics, now it is clear. Now the next question is, what are prebiotics? Any idea?
These are not microorganisms, but they are used in microorganisms, especially for the feed.
Excellent. Ruhul Amin, do you like to add any point on prebiotics?
No, sir, I have no idea. I'm
Uh,
just
pre-
learning.
yes, prebiotics are non-living. Obviously, Shafi rightly said. They are non-living polymeric substances mainly, and they are the feed of probiotics. For example, if you attend a posh, a party in Japan, even a royal party, dinner or lunch, you will see they will offer lots of delicious food items and diverse food items, including raw fish or raw meat and others. And they also offer one dish which is like colorless noodle and a bit slimy, and I didn't understand what are those in the first time I tested, but I found it is tasteless. And our professor, who I thought I saw after taking all the foods, he was eating some of those items. I asked him what are those. He said, "Please use the soybean sauce and take these noodle-like substances." I asked him why, because he said they are polymeric substances, mannan, and they are very good for your intestine. Why? Because they are basically fiber, and fiber is very important for our diet, although fiber do not offer your nutrition, but they are very very important for the probiotic microorganism you are carrying, those who are helping you, because they use these polysaccharide and polymeric substances as their food. So, fibrous, for example, from vegetables, from, the whole grain, wheat, or other grains, you can get lots of fibers and even fruits. And those fibers are basically food of probiotic microorganism living in our intestine, and they are called prebiotics. So prebiotics also, you can understand now, prebiotics also we should include in our diet. And not only in our diet, even if you make a formulation of probiotic for animal or fish or plant, we should add some prebiotic. And those prebiotic would help probiotics to proliferate very fast and exert their beneficial effects to the target organism. Is it clear?
Yes, sir.
Ruhul Amin? Ruhul Amin, am I audible? I'm not sure his connection is fine or not. Ruhul Amin, you are mute. Please unmute and talk. Yes, is it clear that what are prebiotics? Ruhul Amin?
Yes, sir.
Okay, fine. And postbiotics, we shall talk more. Postbiotics as well as symbiotics. There are lots of other terminologies. Let me show you a few slides in this first class. Can you see the slide?
Yes, sir.
Yes, sir.
Oh, very good. So our course title is Probiotics, Prebiotics, and Postbiotics. And this slide is still a bit old. Our institute name is now not Department of Biotechnology. Let me correct it. Institute of Institute of Biotechnology and Genetic Engineering. Okay, so we shall discuss lots of exciting topics on this subject. Let us review the concept of probiotics. The word probiotic comes from a Greek word, and it means for life. Most probably, it was Ferdinand Virgin who invented the term probiotic in 1954. In his article entitled Anti-Wound Probiotica, comparing the harmful effects of antibiotics and other antibacterial agents on the intestinal microbiota with the beneficial effects, probiotica, of some useful bacteria. That means in that paper, he understood and explained that when we take antibiotics, uh, antibiotic first discovered by Alexander Fleming in 1940s or like that. And after that, antibiotic became a silver bullet for treating any microbial diseases. But he had a different thought. He proposed that antibiotic can also kill the beneficial microbes. He named probiotica. So sometime after that, in 1965, Lilly and Stilwell described probiotics as microorganisms stimulating the growth of other microorganisms. So their thought was microorganisms stimulate the growth of other microorganisms. At that time, it was not realized that uh, the microorganism can also, influence the growth of the macroorganism like eukaryote. Many clinical studies have proven the effectiveness of probiotics for treatment of diseases such as obesity, insulin resistance syndrome, type 2 diabetes, and non alcoholic fatty liver disease. Nowadays, there are lots of products available against the obesity. Obesity means to become too fat, and in USA, obesity is a very big problem due to taking junk food and other imbalance food. People can become obese. And insulin resistance syndrome and type 2 diabetes really another emerging problem, and non-alcoholic fatty liver disease can be treated by probiotics. So if you can properly maintain your probiotic in your body, you can keep you free from many diseases. So, how can we define probiotics? To emphasize their microbial origin, Fuller stated that probiotic must be viable microorganisms and must exert a beneficial effect on their host. So, simple definition. Uh, Living organisms, microorganisms that exert beneficial effect on their host. That means they are living in the host. Garner and Schaffsma indicated the necessary use of an appropriate dose of probiotic organisms required to achieve the expected effect. Yes, Shafi already mentioned. In case of probiotic organisms, an appropriate dose is important for having the expected effect. Not only probiotic organisms, in case of anything, dose makes the poison. So if overdose of anything is bad, so even rice is a staple food for us. If you take too much rice every day, you will suffer from obesity and other problem. Even too much water, if someone take, even it would be suicidal. So dose is very important. The current definition formulated in 2002 by FAO and WHO working group experts stated that probiotics are living strains of strictly selected microorganisms, which, when administered in adequate amounts, confer a health benefit on the host. So this definition is highly, acceptable. That means live strains of strictly selected microorganisms, which, when administered in adequate amounts, confer a health benefits on the host. So you have to, be careful in selecting the strain for commercialization or commercial use of any microorganism. The definition was maintained by the International Scientific Association for Probiotics and Prebiotics in uh, uh, you know uh, 2013. So the human gastrointestinal tract is colonized by a complex ecosystem of microorganisms. As I told you, human microbiome project confirmed that an adult human can carry 150 different species of microorganisms. Uh, Intestinal bacteria are not only commensal. Commensalism. Commensalism means not affect the health of the host, but rather uh, exert beneficial effect. But they also undergo a symbiotic uh, co-evolution along with their host. The beneficial intestinal bacteria have numerous and important functions. They produce various nutrients for their host, prevent infections caused by intestinal pathogens, and modulate a normal immunological response. Now, question is, do you think that a balanced probiotic microorganism uh, is helpful to fight against the COVID? What do you think? Shafi, what is your opinion? Do you think that a balanced probiotic microorganism in human body could helpful for fighting against the COVID-19 virus? Or Ruhul Amin, can you?
Yes, sir. Definitely. Since the adequate amount of probiotics, bacteria can exert, they can stimulate immunity, and this immunity can fight against the any kind of viral infection, not only COVID, any kind of viral infection, and the damage, any kind of damage in the or any kind of difficulty in the respiratory tract.
Excellent. Excellent. Thank you so much. Not only fighting against the COVID-19, actually, probiotic microorganisms, recently it has been discovered that they are very critical in homeostasis of our nutrient uptake, as well as they can, trigger the expression of immune genes in the host. So obviously, uh, balanced probiotic microorganism in human health uh, is the indicator of sound health of the host. Modification of the intestinal microbiota in order to achieve, restore, and maintain favorable balance in the ecosystem and the activity of microorganisms present in the gastrointestinal tract is necessary for the improved so the food, synthetic food or junk food we are taking, sometimes they are stressful for the, ecosystem of your intestine. Obviously, the microorganism living there. So, the modification, you can modify them and also help them to how to say sorry help your intestine uh, uh, in a good condition so that you can keep you healthy. They may be consumed in the form of raw vegetables and fruits, fermented pickles, dairy products, and another source may be pharmaceutical formulas, and functional foods. Now, the question is, how can you modulate your intestinal microflora in a desirable state? How can you do it? You can take raw vegetables, because raw vegetables as salad or fruit, uncooked, obviously they are. Fermented pickles contain lots of beneficial bacteria which are probiotic. That dairy products like yogurt and cheese,
Yogurt, ghee,
yes,
cheese,
yes, they can.
doi.
Yes, though, yes, card. So they are the source, and you can consider during your diet, so that you can regularly take some vegetables, fruits, or dairy products like curd or cheese or like that. So, probiotics are not only in human, they are present in all other eukaryotic organisms. Probiotics are mainly, bacteria, bacteria like, you know, different kinds of bacteria. But in case of human, lactobacillus and bifidobacteria, they are the predominant, but there are many others. So bacteria are they are probiotic organisms, and they are actually prokaryotic organisms. So in intestine, here you can see the analogy is that lots of probiotics are there. In case of plant rhizosphere in the root zone is called rhizosphere, also is the harbor of huge number of diverse probiotics. And without microorganism, you would be wonder to know that without any probiotic organism in the rhizosphere, plant cannot take a nutrient from the soil, because a plant can only uptake nutrients, 16 or 17 essential nutrient elements in ionic form. For example, nitrogen, phosphorus, potash, all are in ionic form. And naturally, a very small portion of these nutrient elements are available in the ionic form in the soil. So microorganisms, by through mineralization processes from organic and inorganic sources, they break down the organic and inorganic materials, and then offer the nutrient in ionic form to the plants. Why they do it? Because they also need and uptake the nutrient in ionic form. They use different enzymes to break down the complex organic and inorganic materials, which contains different which are the reservoir of different nutrient elements. And other point is that plant in the earth, plant is the only organism which has the a unique biochemical process called photosynthesis. They can trap the solar energy and convert into chemical energy, and a large proportion of the chemical energy, nearly 40%, they release through the excrete through the root, so that microorganism in the rhizosphere can get the energy from the plant. And this is why it is a mutual and symbiotic relationship. Microorganisms work for them and offer the nutrient in available form. So you can imagine that, like our intestine, rhizosphere is very important place. If we can modulate the microorganisms present there, if you can enhance the beneficial microorganism over there, you can promote the plant growth, as well as some of the microorganisms living in our intestine, or on the in our body or plant body. They can produce different antimicrobial substances to ward off pathogenic microorganisms. They are potential to infect the host, because they care the host. So these are the fundamental points which we consider for the usefulness of the probiotic microorganisms that we can discover from the plant or animals or even in human. In case of plant, biofertilizer, biopesticide, or biostimulant, they are now big industry, billion-dollar industry in the world, and the growth rate of those industries are 10 to 15%. So this is really amazing. I think you can see the relationship between different organisms, how they are interrelated. So I would like to stop my talk here today because this is the first lecture, and I guess you know our university system, uh, the examination system. We have the provision to take a quiz exam at any time. We have two midterm exam. Quiz exam 10% marks. There are also assignment or project work, 10% marks, and 25% marks for each midterm exam, so, and 30% marks allocated for the final exam. During COVID-19, we are now, till now, allowed to, evaluate you for the quiz, midterm, as well as the assignment. So this is all about today's deliberation from my side, and you are welcome to ask question.
স্যার এই কোর্সে তো so far I know Mahbub sir also included in this course. So both of you will conduct, or who will conduct majority of the class?
And the probiotic, this BTL 611,
Yes, sir.
I shall conduct this course, and Dr. Mahbub may come in a point to deliver a talk on uh, probiotics in aquaculture.
Okay, sir.
So, any other question? Shafi, have you understood all the points? And how do you feel coming back to the school after a pause? Please unmute yourself. Yes.
Yes, sir. It's also nice and interesting topics for me as well, because it's also I know a little bit, but we can learn more from this subject.
Okay, thank you. And we often organize Zoom meeting for our research team. So you are two new members. Uh, Nuruddin Mahmood usually coordinate the meeting. So you will be welcome. And good news is that we are uh, maybe early next year, we shall receive one postdoctoral fellow, Dr. Paritosh Kumar Rai, and also another master student from, I'm not sure, Maulana Bashani Science, Bangabandhu Sheikh Mujibur Rahman Science and Technology University. So our team is increasing day by day, and although COVID-19 has restricted lots of our activities in the campus, even though our laboratory, IBZ laboratory, we are still keeping it live, and not a single day we stopped our laboratory activities. Some of our dedicated students, they are working very hard, and they produced lots of data during the COVID-19, and we published nearly 30 articles in the scientific journals and books. So our group is highly productive, and I am expecting both of you highly enthusiastic student who would make significant contribution to our journey. And we are getting lots of new, funding as well as collaboration. Hopefully, you can enjoy and you can find opportunities to prove yourself and obviously to do some groundbreaking research work. Our sequencing machine recently sequenced the whole genome of jackfruit. We are now working hard to assemble the small reads and announce the genome sequence. That would be a landmark. So think how can you utilize that fantastic facilities, because in our public universities you cannot find such a great Illumina sequencing platform. So it has just started its journey. So you can have the opportunity to use Illumina sequencers. We have two sequencers, Express 550 and Minisec. I am expecting something new. And although COVID-19 is going on, but I shall expect by the end of this term, you shall start, going to the laboratory. Even if you want to start research uh, 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 earlier than my target, you can do it because some of our students are working there, and we can move on. A part of your time you can invest for your research, and obviously adapting to the must coursework and do well. There is obviously a big target in the first term of masters or PhD program. I thank you, both of you. Any question from your side, or comment, or any expectation?
Sir, how can I understand the bacteria, whether it is probiotics in case of in laboratory?
Pardon?
Can I repeat, sir?
Yes.
Um, uh, we have today studied the probiotic bacteria mainly. So, how can I understand in a laboratory that this bacteria is probiotics? Uh,
In case of plant, it is very easy. Within a week, if you apply probiotic bacteria and colonize it, and in the petri dish, on water, soaked filter water, if you allow to germinate the seed, you can see how robust probiotics on growth and germination of the seedling, or seeds, germination of the growth of the seedling. But in case of fish, we have some experiment in the aquarium. Dr. Mahbub, he has been lots of research work on the effect of probiotic bacteria on the fishes. But in other animals like cattle or
Poultry.
poultry, we have not yet tried. But in that case, what you do, in case of poultry, you have to isolate the probiotics from the guts of the poultry, and then you apply them through the feed, like we offer to the fishes, and then see the effect. It is possible in case of poultry, but in
Okay, sir.
that case, you have to target, you have to isolate the your
Bacteria.
bacteria first and check whether they are really probiotic or not. We have the mini set, so you can use the 16S RNA sequencing, identify the bacteria, check the uh, gene bank whether it is really probiotic or not, and then you test the efficacy in the animal system. Other point that even one of the interesting subject would be, we call deshi murgi or
Yes,
broiler,
Sonali.
deshi murgi, broiler, shunali. And in the market price, there are huge differences. If you check the gut microbiome of deshi murgi or shunali or the broiler, you can get the some after our metagenomic analysis, you can get understanding the differences of the microbiome that is probiotic bacteria in their guts, and you can make some correlation which bacteria are playing the role. And then you can target those bacteria from the deshi murgi and then apply them into the broiler or sonalika to get better smell of the meat as well as taste of the meat or like that. So there are lots of area to explore and publish in high ranked journal, and we have all the facilities available in our IBG.
Thank you so much, sir.
Shafi, do you have any question? Please unmute yourself.
No, sir. I think everything is fine.
And I expect, Shafi, please think in your mind what kind of research you can do in future in our laboratory. Because in case of PhD research, I expect by the end of this term, you will start your job slowly. Because 3 years you can see time flies, and 3 years is not a very long time for doing some groundbreaking work. So you can discuss at any time whenever you like, and I shall welcome you. Thank you so much,
Okay.
then,
Okay, sir.
and
Thank you.
have a good day. And you have another course today, yes,
Yes, sir.
at 2 o'clock, right, Shafi?
Yes, sir.
Then see you at 2 o'clock. And Ruhul Amin, have a good day.
Wishing you a good day and warm welcome to your birthday. That may Allah keep you sound and safe and give you more success in the area of research and helps to uh, guide all the student and the researcher, enthusiastic researcher in Bangladesh. And I wish that may Allah make you or give the chance to become a vice chancellor of any university in the Bangladesh,
Okay.
because because the
And,
background
thank you.
back,
Actually, our student always celebrate my birthday
yes sir, I know.
in COVID-free situation. And this year it is a special situation. So I am receiving the electronic messages from lots of people in the Facebook Messenger. Anyway, thank you so much for your good wishes. Vice Chancellor is not the only target to do deliver good job for the nation. But anyway, I thank you for your wishes. All the best wishes and have a good